This is Chapter 2, Lecture I on proteins, and proteins are incredibly important to our body. In fact, proteins compose about 10 to 30% of the entire cell mass. They perform a lot of different functions. They can play the role as enzymes, and enzymes are the biological catalysts. So one of their big functions is that they lower the activation energy, and the activation energy is abbreviated as E with a subscript of A. So I want you to watch the videos on enzymes and mastering A and P, as this is one of the most important functions of proteins in our body. Proteins are also going to work as construction materials all throughout the body. So they're going to help to make plasma membranes. We'll talk a lot about them and their specific functions in Chapter 3. They're also functional proteins, and one of the most important functional proteins that you see on this specific slide right here is that of insulin. And this shows the specific amino acids that are going to make insulin. And amino acids are the very important building blocks for proteins. And all proteins are going to be composed of many important elements. For example, there's carbon, hydrogen, oxygen, nitrogen, and there's also sulfur as well. So this slide is showing the proteins. Again, there's carbon, hydrogen, oxygen, nitrogen, sulfur, and sometimes even phosphorus. Proteins are going to be polymers. They're very, very large examples of proteins, and they're all made up of the building blocks, which are amino acids. And there's 20 different types of amino acids, and again, these are the monomers, the small units that make up the proteins. And these amino acids, as you see on the screen, they're going to be made up of an amine group. The amine group is the NH2 group, and also the acid group, which is COOH. So those two are the main groups of amino acid, or the two main groups that make up amino acids. They're joined by covalent bonds, which, remember, occur when electrons are shared. And the other important group is this box that's in the green on this figure, and this is the R group. The R group is going to differentiate. It's going to be different between various amino acids, and it's what really determines the nature of the specific amino acid. So when these amino acids are put together, then they're put together in the same manner in which carbohydrates are put together. There is dehydration synthesis. So again, this is an important example of chemical reaction you need to know. So dehydration means the removal of water. And with the removal of water, you can see that there is a bond that is formed in the products. And this bond here is called a dipeptide. A dipeptide is going to be made up of two amino acids that are put together. There's also a tripeptide. As you would imagine, the tripeptide is made up when there's three amino acids not 33 amino acids, but three amino acids that are put together. This is going to make a tripeptide. And then when we have greater than that, when we have greater than 10 amino acids, this is going to make a polypeptide. And most proteins have just thousands of amino acids that make them up. And hydrolysis then is the opposite of dehydration synthesis. This is used to break down a larger molecule. For example, dipeptide is broken down to two amino acids by adding water. So remember, in the body, all these chemical reactions are actually reversible based on the needs of the body. Our next slide is showing the structural level of proteins. And when we consider the structural level of proteins, there are four different levels. There is the primary solution, or the primary structure. And the primary structure is really simply the amino acid sequence. And so that's what you see at the top of your slide. Primary just means the amino acid sequence. And in this case, it's actually simply a linear structure. So it's the simplest type of structural organization for a protein. And proteins in our body are going to be a lot more complex than this. The second structure, the secondary structure, is going to be either a beta-pleated sheet or it's going to be an alpha helix. And in both cases there are hydrogen bonds, which you've learned about in a previous mini lecture. There are hydrogen bonds that connect the primary structures. So the hydrogen bonds are going to be important for sort of stabilizing this structure. Then we have the third, which is the tertiary structure. And in this case, there are alpha and beta sheets that fold on top of each other. So these alpha and beta sheets, they connect on top of each other. And in this case, we end up seeing a so-called globular protein, so a more complex protein. And then the last one is really the most common. This is called a quaternary structure. And in this case, we end up seeing more of a functional protein. And there are 2 or more polypeptide chains. So 2 or more polypeptide chains in this case. And hemoglobin would be an example of this, which would be a quaternary structure. Again, these are the most common. Your next slide is going to be the same figure that you have in your textbook. This is figure 2.19. And the first one is going to show the amino acid structure. So you can see again, this is the simplest. It's a linear structure, so it's really just a chain of amino acids. The next level is going to be either the alpha helix or the beta pleated sheet, which almost looks kind of like an accordion. And again, these are going to be stabilized and connected by hydrogen bonds. And again, as you go through the chapter guide in Mastering A&P, you're going to see links to different animations. And again, I really want you to look at these animations. They're very helpful, and they're for the primary and secondary structures. The next slide is showing the tertiary and quaternary structures. The tertiary structure occurs when there is a superimposition on the secondary structure. So imagine 2 or more secondary structures that are actually folded on top of one another. And again, this is what's going to make a compact globular molecule. So that's really a hallmark of a tertiary structure. And so an example of this would be the tertiary structure of prealbumin. Albumin is going to be an important plasma protein in our blood that transports other molecules, and it plays a role in osmotic pressure. In this case, it transports the thyroid hormone, thyroxine in the blood and cerebrospinal fluid. Then the quaternary is the most common, again, and it's the functional protein, so it has functional properties. And in this case, hemoglobin would be a great example of this one. And there's no L in hemoglobin. But again, I want you to look at these animations that are in Mastering A&P. Mastering A&P is going to be your best friend, not only this semester, but also next semester. So these proteins can be construction molecules in the body, so they serve as very structural proteins, and some only exhibit a secondary structure. Many of them are going to be tertiary and quaternary, but they're going to use mechanical support. They're going to provide tensile strength. So some of the best examples of these would be collagen. It's the single most abundant protein in our body. Also, keratin and elastin would also be examples of structural proteins that are very, very important in our body. And you can see a zoomed-in electron micrograph picture here of the hair shaft, showing some of the collagen and elastin. These are 2 main components of connective tissue. And in Chapter 4, we're going to talk about specific types of tissues. There's a lot of examples of connective tissue. Next slide is showing globular or functional protein. So the word globular means functional, and the word fibrous means structural. So notice that this is described for you at the top of the slide. So globular or functional proteins are those that are going to, again, be tertiary or quaternary in structure. Again, a lot of times they're going to be quaternary as well, but the important thing to remember here is they're going to have a specific function. So they're going to function as antibodies, as hormones, molecular chaperones, which we'll talk about in a couple slides, and also as enzymes, which are very, very important. And the thing I want you to notice is that these enzymes specifically have what are called active sites. The active site is the region of the molecule where the chemical reaction is going to happen. So these enzymes, remember, all act as biological catalysts. So they're going to lower the activation energy. They're going to ensure that a chemical reaction happens much more quickly. And we need these for survival. They're very, very important in our body. Now, protein denaturation is a very serious problem, and this is one of the reasons that the body has to maintain a certain pH level. So remember, the pH level for our body needs to stay within 7.35 to 7.45. So if this pH even gets slightly out of this range, then there could be the damage of proteins, and that could be very, very, very serious. So an irreversible damage, an example of this is when you cook an egg, it actually changes its nature into something else. So in the body, the pH needs to be maintained as well as the temperature needs to be maintained at 98.6 degrees Fahrenheit or 37 degrees Celsius. So when this changes even a little bit, then that can denature very important proteins. So enzymes could not form. There could be incorrect folding of a protein. If a protein needs to be in a quaternary structure, it's never folded to where it needs to be to perform its function. So very, very serious. Our next slide is on molecular chaperones. And you all know what a chaperone is. A chaperone could be a parent that goes on a school trip with a son or daughter and helps to protect and watch over the other students. And so in this case, a molecular chaperone is going to protect a protein so that it functions correctly. So it protects a protein so that it functions like it's supposed to, functions correctly. So it's an additional protein. It's going to assure accurate folding and association of other proteins, prevents incorrect folding, and one good example of this that I can mention is when I was in grad school, we did an experiment where we studied stress proteins. So a stress protein is going to protect a protein. It's going to protect a protein from excessive stress. And we have these in our body because we all deal with stress every day. And it's very important that we have something present, a molecular chaperone, that is going to protect from excess damage. So in this case, on the figure that you see on your screen, you see what's called a heat shock protein. This heat shock protein is going to protect a protein from excessive heat buildup. So in order to test this in the lab, what we did is we used the little pill bugs that you're probably familiar with. Sometimes they're called roly-polies, but these pill bugs are going to release heat shock proteins, and this heat shock protein in this case is abbreviated as HSP. They're going to release heat shock protein when stressed. So what we did is we stressed these pill bugs, we dropped them in liquid nitrogen, and then we measured the heat shock protein production before and after they were stressed. So after they were stressed, we found that there was an increase in the heat shock protein, as you would imagine, once they were stressed. This is exactly what's going to happen in our case. The heat shock protein is going to protect important molecules. In this case, the heat shock protein is going to protect the DNA so that it can't be damaged by stress.